Dr. Derr with Texas A&M University last year. I don't see him. I don't where. There he is. So his last presentation is on YouTube, actually. It's really like incredibly, amazingly interesting, this study that they did on bison, which he talked about last year. But this year he's going to be talking about wildlife genetics, which was similar to what he talked about last year, but it was specifically with bison. So I'm going to go ahead and bring up Dr. Derr. He's incredibly bright, intelligent, smart, charismatic, and he's a wonderful professor at Texas A&M. So let's welcome Dr. Derr.
I think I have this, yeah. I want to thank Brian for the invite to come here today. I teach at Texas A&M. We now may or may not know this, but the College of Veterinary Medicine's name recently changed at Texas A&M. Our university president demoted us to the School of Veterinary Medicine. So it's kind of hard for me to get used to saying School of Veterinary Medicine when I kind of used to the College of Veterinary Medicine for 30 years now. But at the School of Veterinary Medicine, I teach a class. I teach it for pre-med, pre-vet students. It's called Biomedical Genetics. And I have about 110 students in this class. They all want to go to medical school. They want to go to vet school. They're all real aggressive students. They're juniors and seniors. You kind of know the type. Some of you probably have some children that are like that. They're real aggressive. They know what they want to do. They're very smart. They work really hard. Most of them are classic overachievers. And yeah, there's probably one right there. And I'm giving a final exam on Tuesday in this class. And it's very nice to get out of College Station. I didn't tell any of the students that I was going to be gone this weekend. I'm not going to answer my phone. I've got 2 messages since we sat down here this morning. Students have an immediate question they need an answer to. So it's a pleasure to be here. It's fun to talk about something I really love to talk about, and it's fun not to have to deal with those students for a little bit. But I have to go back to College Station later today, so I'll get to see them. So I'm a geneticist. I do wildlife genetics research. In the past, I used to do a lot of livestock genetics, but my real passion was wildlife. And so I do wildlife genetics research here in North America. I do some work in Central and South America, and I do some work in Africa. And I think my objective today is similar to what it was 18 months ago when we met here, and that is being at the College of Veterinary Medicine or the School of Veterinary Medicine is a little bit different mentality than, say, for example, if I was over in biochemistry or if I was in biology. In veterinary medicine, we're very, very practical, okay? Because we're dealing with sick animals, we're dealing with injured animals, we're dealing with problems that need solutions now. It's not necessarily academic, it's not necessarily theoretical, but it is finding solutions to problems, okay? That's what veterinary medicine is, trying to solve problems. We come at genetics with the same mentality, and I do too. I'm not too interested in basic research. I'm interested in applied research, and I'm interested in how we can use this information, how we can use this tool of genetics to learn things we can't learn any other way, and to make significant improvements in things using these tools. So that's kind of what I want to do today, and that's kind of the plan. I'll talk a little bit about genetics and genomics. For the most part, you guys have a definition of those 2 terms. I'm going to give you my definition as a geneticist, and how we're using these tools to be better stewards of wildlife. I gave 2 examples actually at our last meeting 18 months ago. I'm going to give those same general examples today. They're great, I think, examples of how we use biotechnology and genetics to learn things we can't learn any other way. I'm going to highlight some areas where our lack of knowledge and our ability to use these technologies with exotic wildlife is a problem. It's a serious problem. We are so far behind in wildlife genetics compared to livestock or human genetics that it's almost insurmountable to catch up. But what that does is it offers us a lot of opportunities, and I'm going to point out some of those opportunities today. And then finally, I want to lay out a challenge for us, and that challenge is to think about ways that we can develop these scientific tools to take advantage of these technologies for exotic wildlife. There are certain species of exotic wildlife that we have a lot of genetic tools for. The charismatic ones, you know, the elephants and the rhinos and the buffaloes, we have a lot of tools for those. But the other animals, and many of the ones you guys raise, a lot of the plains game things from Africa and around the world, we don't have a lot of tools for those species. And there's no central clearinghouse where you can go take advantage of these technologies. So to find the science of genetics and genomics just a little bit, I'm going to give some advances of how genetic technologies are being used with wildlife. I, for the last 25 or so years, have been doing North American bison genetics research. I've had 5 PhD students complete their dissertations with North American bison, so I've got a lot of experience with these things. I've collected DNA samples from virtually every major bison herd in the world, from the United States, Canada, Mexico, to Russia and Poland. And so I have a lot of experience with bison, and I have a lot of great stories about bison, and I think that more than any other species, really, bison are a success story for conservation biology. We all know what happened to bison 120 or 30 years ago. We all know what happened, from millions to hundreds. No other population has suffered population crash of 99.99 or more % of that population, and then recovered in less than 100 years to the point that we could have bison burgers for lunch today. I mean, what's another species that that's happened to? So from a genetics point of view, my interest is, why did bison recover so well? Why were they so successful in their recovery? I'm not going to spend a lot of time talking about today that subject, but I just wanted to give you some background as to why I'm so high on looking at bison as a genetic tool. And then I'm going to talk real quickly about a study I did for Safari Club International with Sitka deer. It was interesting. It was unusual. The findings we found I would not have predicted, and it changed the way Safari Club does business today. And then finally, like I said, I want to talk about the current resources for exotic, non-native wildlife and talk about some of the needs and opportunities. So genetics, we all have a definition of what genetics is. I hear people all the time using the term genetics. This guy's got really good genetics. This guy's got bad genetics. This guy's got an inbreeding problem. This guy's got some other genetic issue. So let me just define from a geneticist standpoint what these terms mean. Genetics is the scientific study of heredity and inherited traits among individuals. A geneticist is interested in what gets passed on to the next generation. That's what I do research with. I am very interested in what goes from one generation to the next, whether it's from one individual to their offspring or from one cell to the daughter cells. In both cases, that's moving genetics from one position to another. Wildlife genetics is the use of molecular tools to determine parentage in many cases, to determine inbreeding, genetic diversity, in some cases ancestry, individual identification, and specific traits of interest such as body, horn, antler size, disease resistance, fertility, and so forth. Genomics really focuses on the structure of the genome, how that genome is put together, mapping genes of importance in these genomes, and we use genomic technologies as a tool to determine where genes are in the genome and what those genes do. Genetics and genomics are powerful tools for managing wildlife populations. And some groups in the Safari Club International have developed a position statement regarding the use of these technologies. And I think this position statement is important because I want to be clear what I'm talking about here with genetics and what I'm not talking about. You can read the statement here from Safari Club. Basically, this was done a few years ago. This was from the Record Book Committee when they were talking about what animals, what species to put in, new species to put in the big game species in the record book and for World Hunting Awards. And the decision was made that all SCI record book entries would use the best available science regarding the taxonomic status of an animal. SCI Record Book Committee will add new species to the record book based on scientific evidence that that entry represents a valid taxonomic species or grouping of related subspecies and not simply a hybrid, which in some cases has happened, or a simple color variant or genetic mutation of an existing species. SCI Record Book Committee does not support procedures and practices in wildlife that produce non-typical color variants, horns, antlers, and body size. I know this is controversial, certainly in Africa with a lot of different species it is, but this is what Safari Club decided they were going to do. And finally, maybe more importantly, the Record Book Committee discourages breeding practices that genetically manipulate wildlife species to alter appearance and size, including assisted reproductive technologies that include genetic manipulation. I do genetics work. I am involved in some genetic engineering technologies, not for wildlife, but in other species. And I'm not a proponent of genetic engineering technologies for wildlife. If you think it's not a big deal, you will most likely find out that it can be. Because when things are possible, there will be people that want to do them. And putting genes from one species into another species to cause a different kind of trait is going to be a problem in this industry. Maybe not next year, maybe not even in 5 years, but trust me, it's going to be an issue. Because the genetic engineering technologies that have been developed just in the last 3 or 4 years, just since COVID happened, are amazing. There soon, and I predict within the next year, will be genetic engineering technologies that are available for people to cure certain single gene traits in people. Not in embryos, not in even newborns, even in adults. These technologies are very well advanced. They work really well. They're fairly simple to do. And I'm just giving you guys a little warning right now. It's a good thing, I think, if it's used correctly. Very good thing if it helps people who have genetic diseases. But it's got the potential for a really serious downside. Okay, so let me give you a couple of examples of what I think are really fun projects that we've been involved in using genetic technology for wildlife. Now, you may have heard this story before. I've added a little bit to it from last time, and I just think it's a wonderful story that I like to tell people about how we use these technologies for a wildlife herd. If you've been to Wind Cave National Park in South Dakota, you know how special a place it is. It's an extremely special place to the Lakota Sioux. That's where they feel like they came from, out of the caves at Wind Cave. They were led by a white bison that came out of the caves there. This white bison then turned into white buffalo woman, and that's where the Lakota Sioux feel like their origins were. So this is a pretty sacred place. This is a pretty special place for bison, and it's really a special place to get to work. There at Wind Cave National Park, they select on 2- and 3-year-old calves, and they decide how many they're going to keep in the herd at that age. The rest of them, then they disperse out of the herd, so most of them are sold or given to Native American tribes. But they keep the sex ratio at 1:1, so it's more of the kind of sex ratio that a bison would historically have that would not necessarily be in such a managed herd. In 1999, I went there with a graduate student. We had a project with the Department of Interior. We sampled the entire herd, got DNA samples from everything at their roundup. Then I went back pretty much every year, or sent a student back every year for the next 10 years, and we also collected DNA samples from their calves. I didn't have really any money to do parentage testing on those subsequent years, but I had enough money to go to collect the DNA samples and to bank those DNA samples. So we already had all the adults, and we already had parentage markers for all the adults. We just collected DNA from all the calves for 10 years. And then at the end of that time, in about 2010, I got a grant from DOI because I had the DNA samples, and that's going to be a topic I'm going to come back to in a few minutes. I had the DNA samples, so that was the reason I got the grant to parentage test every one of those calves for 10 years. No one had ever done that. No one had ever looked a 10-year period of time in a herd population to see who was doing the breeding. Our findings were, I think, unexpected, certainly not what I expected, and surprising, and we learned a lot about wind cave. So we asked a whole lot of questions. I'm not going to go into the entire study. Some of the questions were on the mom side, some of the questions were on the bull side. But for example, how many bulls produce calves each year? Okay, so there's about 350 to 400 adults in this herd. How many bulls, and it's 50-50 sex ratio, how many bulls are producing a calf every year? That seems like a reasonable question. How many bulls never breed? How many bulls were in this herd as an adult animal through this time period, didn't produce any calves? How many bulls produced, or how many calves were produced by each bull? You know, were there bulls that produced 2 calves or 5 calves or 20 calves in a given year? And what's the age of the bulls that produce calves? Back then, if you ask any bison producer, and some big bison producers, who are your major breeders? Everybody pretty well knew 6-7-year-old bulls seem to do most all the breeding. They would had people tell me up to 60-70% of our breeding is probably 5, 6, 7-year-old bull calves. They do the vast majority of the breeding. Okay, so we looked at a lot of parameters here. I'm just going to show you a couple of the ones I think are kind of cool. Total number of offspring produced by individual bulls. Just between the years 2002 and 2006, 107 bulls produced 280 calves. Over this time period, there were approximately, on average, about 175 mature bulls in the herd. You can look, you know, some of these bulls produced quite a few offspring, but not very many. Most of the bulls that produced offspring only produced 1 or 2 or 3 calves in that time period. So I had a graduate student, her name is Natalie Halbert. She's still around. She's not my graduate student any longer, she graduated, but she came in with data from 2000, 2001, one day to my office and said, I have some interesting results. I said, what's that? She said, when I look at the age of the bulls that are producing calves in the herd, some of these 1.5-year-old bulls and some of these 2.5-year-old bulls are producing calves. And then, as you could predict, 4, 5, 6, and 7 go up. And I said, well, we need to get the rest of the years to see if the data are consistent, if we continue to get that. She did. We got it for all 10 or 11 years. And this is just kind of a synopsis of what she found. She found that about 10 or 12, somewhat percent of the calves produced each year, on average, the sires are 1.5-year-old bull calves. And the 2-year-old bull calves don't produce as many, but they produce some calves. And then, really predictably, 3-year-old bull calves don't produce very many calves. What the heck is going on? I had no idea. And this student, Natalie, was a lot smarter than me. And she said, I think I know, and I think I know how to test it. She said, let's do a genetic relationship test on the bull calves, these young ones, and see if they are producing an offspring with a female relative. And she did. And almost inevitably, these 1 and 2 year old bull calves, the dam they're producing a calf with is a first or second degree relative. So a sister, or half sister, or cousin, or aunt. What in the world is going on? If you spent much time in a bison herd, it's pretty obvious. When the calves are born, they stay with the matriarchal groups, and they stay with them and they protect the calves. The bulls generally, after breeding season, go off and do their own thing. But the young calves, males and females, stay with the females. When they're there, they're doing some breeding. But by the time they're 3 years old, they get pushed out of those matriarchal groups. They can't compete with the big guys for the breeding, and they can't get back into the matriarchal groups because those females won't let them. They run them off. You drive around Wind Cave during breeding season, you'll see that there are these herds of 3, 4-year-old bull calves. They're similar to junior high boys. Nobody likes them. You want them to stay, you want to stay away from them. And they're causing trouble, they're fighting with each other, but they're not doing any breeding with anybody. So think about that for just a second. There is a built-in this bison herd, inbreeding that goes on. Somewhere around 15 to 20% of the calves are produced by relationships between relatives. That has significance for how it changes the genetic architecture of a bison herd. That is a built-in breeding mechanism that was not determinable without genetic technology. So this is just a kind of a synopsis of what we found based on that information. It has changed how bison are managed in national parks today. And it certainly has changed how bison are managed in many large private herds. There are some private bison herds that have more than 5,000 animals, and much more than Wind Cave National Park. And the managers of those herds are very interested in the conservation of genetic diversity over time in those herds. They do not want to lose genetic diversity. They're not going to have a sex ratio 1-to-1 in a commercial herd. That's not going to happen. It's going to be 5-to-1 or 10-to-1 or 15-to-1 even in some herds of females to males. But they still don't want to lose genetic diversity. So what many of these populations now have decided to do based on this study is that they retain those bull calves until they're 2.5 or so years old. And then those bull calves go out of the herd, go to feedlots, get fed out, and become meat. But they leave them in the herd so that they can put their genes back into the herd before they depart. That's important because what that does is it raises the effective population size. That's a term we use in genetics for how many animals breed each year, each season. It raises the effective population size and it preserves genetic diversity in that herd. Inbreeding is not always bad. If it wasn't for inbreeding, we wouldn't have breeds of cattle, we wouldn't have breeds of pigs, we wouldn't have varieties of wheat and barley and corn and soybeans. All that happened because of inbreeding. So you can take advantage of inbreeding in some cases, and that's what bison herds are doing, and that's what some national parks, some fish and wildlife refuges, and a lot of private herds are doing today. So one other example, I'll just go through this example pretty quickly. The SCI record books folks approached me. This has been a few years ago now. There was an animal that was submitted for the record book. It was a pretty incredible animal. It was submitted as a Sitka deer. There were some committee members, including Gary Rose. A lot of you guys probably know Gary Rose, worked for Dan Duncan over at Smithville. very knowledgeable person, and he had some suspicions that this animal actually may not be completely a Sitka deer. So they approached me and said, is there any way you can tell if this animal is a Sitka deer or not using genetics? And I said, potentially. I don't have a lot of genetic resources for Sitka deer, but I'm happy to take a look at it, hoping they had some meat or they had some hair follicles or something I could easily get DNA from. As it turns out, they did not. It was a shoulder mount on the wall at this point. It was tanned leather. So we were able to take it off the wall, cut some of the hide off the back of it, finally isolated some DNA from that. It's quite difficult to do, but we did. So the first thing I did, you want to do the simplest thing first. The first thing I did was simply sequence a region of the mitochondrial DNA. So you guys all remember from high school biology, we all have mitochondria in our cells. That's where our energy is produced. And there are genes in the mitochondria. There's DNA there, and there's genes. And every one of us have the mitochondrial DNA from our moms. It comes with the egg cell, it doesn't come with the sperm cell, okay? So you females can pass your mitochondrial DNA off to your sons and your daughters. You males, you're an evolutionary dead end when it comes to mitochondrial DNA because it doesn't come with sperm cells, okay? So we sequenced the region of the mitochondrial DNA that is generally species specific. We sequenced it, we looked at it, we compared it. My hope was that it was not Sitka deer, it was red deer. If it was red deer, that's the end of the story. This sucker's a hybrid, and that's that, okay? It's fairly easy to do. I run a DNA sequencing lab. We did it. Making DNA was the hard part. Doing the analysis was the easy part. Got the DNA sequence, compared it against Sitka deer, compared it against all known mammals. It was a 100% perfect match Sitka deer that had been published. I'm like, So this animal's got Sitka deer mitochondrial DNA. It was submitted to trophy records as a Sitka deer. Is that the end of the story? Well, remember, we're only looking at the mom side of things, okay? Only the mom's mitochondrial DNA. So it could still be a hybrid. It's just got an unbroken mother, grandmother, great-grandmother, great-great-grandmother that's a Sitka deer, okay? So I didn't have any other resources to do nuclear genes. I only could do mitochondrial genes at that time. There was a paper, though, that was published in the UK looking at a whole lot of different cervids, red deer, fallow deer, Sitka deer, and other things for some nuclear markers. I contacted the researcher in the UK. I told her what we were doing. She said, send me the DNA. I'll see if it will work for our markers. And so I did. I sent her the DNA. She looked at some nuclear microsatellite markers. And as it turned out, this animal had a number of nuclear gene markers that never had been seen in Sitka deer. In Sitka deer in Japan, in Sitka deer in Europe, in Sitka deer in the US, had never been seen in Sitka deer, but were common nuclear markers in red deer. But it wasn't 50-50 like a first-generation hybrid would be. It was about 30% red deer and about 70% Sitka deer, not a first-generation hybrid. So we know some stuff. We know it's got an unbroken female lineage, the Sitka deer. We know it's not a first, and that could be a Sitka deer female, a red deer male produced this hybrid. Then it would be 50-50 on the nuclear side, but 100% Sitka. Wasn't that. So this was a subsequent generation, second generation, third generation, maybe fourth generation, since the original hybridization event. As a matter of fact, I think this was a purposefully made animal. So we completed the study. I went and gave my results to SCI Record Committee. They looked it over. Actually, The place, this was killed on an estate in Iowa. As it turns out, the owners of the estate didn't know anything about it. This was an animal that was put there. This guy was recruited, paid a lot of money, came in, shot this animal, left with it. The guys he dealt with are nowhere around. The owner, the sport hunter, was there. I felt pretty bad for him because he had been duped. He's got a trophy, and the trophy's on his wall, but it's not in the record book, and it was denied by record book. This is how you can use this technology. So here are just two examples, one with bison, learning about breeding, one using this technology to learn about the taxonomy and the breeding history. And my point here, what I'm trying to argue, is this technology is cool. We can do things with this technology you can't do any other way. And it's fun to do it. It's almost like investigative reporting. You know, it's almost like forensic analysis of something. It's easy to get students motivated, and it's easy to do this kind of work because it's kind of cool in the end, because you have something that is usable. Worldwide, we don't have a lot of genetic tools for wildlife like we do with livestock species. I work at Texas A&M with a lot of livestock geneticists. Okay, some of my best friends are cattle geneticists, swine geneticists, poultry geneticists. I look at the tools and the resources these guys have, and I look at the money these guys have to do this. It's pretty amazing. We just don't simply have that kind of stuff for wildlife, at least that's readily available. Parentage tests and some diversity ID tests are available for some species here. There are other species too that these things are available for. White-tailed deer, there's a lot of resources. Mule deer, there's some resources. Elk, there are some resources. But when you move away from some of those species and you get into more minor species and a lot of the stuff you guys deal with here that are exotic species, it's hard to come up with these resources. They're just simply not available. Most US-based exotic species have very limited genetic tools available to them for off the shelf. So after I was here 18 months ago, probably had five or six people, some of you guys may be in the audience today, contacted me and said, I need some parentage information from, name the species, black bucks or impala or springbok. And it's very hard to have off-the-shelf kinds of tools where I can say, sure, send me the DNAs. I'll be happy to do a parentage test for you and tell you, who's the daddy. It's hard because we don't have those resources off the shelf. I have to go out and try to find those resources, find someone at Stellenbosch or find someone at University of Pretoria or find someone at a company here in the United States that does that kind of testing. I say we need to change that. I say we need to develop some technologies so that we can start doing the kinds of studies that I just laid out here for you. And I have about 200 more examples of how we've used genetic technologies to do really cool things. If I get an opportunity to come back in a year or two when you guys do this again, I'll give you some other examples of how we're using this technology, because there are lots of opportunities there. But we have to have the resources. So from an academic standpoint, if I'm going to step back and say, I want to develop these resources for the 20 most common species that you guys raise that are exotic species, and I want to develop these resources where we don't have them today, then the way it would happen, the ideal way it would happen, is that I'd have enough money that I could get 50 or so individuals, some that are here in the US, some in their native habitat, so we know what's there too, at least 50 animals, sequence their genomes, their entire genome, compare those genomes, develop a set of markers specific for that species, array that on a microchip, and provide that as a service for that species. We're talking about a project that's a PhD project and $100,000 to do it right. That's not going to happen. No one's going to give us that kind of money to do that in a species that's economically doesn't make people that much money. It's been done for rhinos, it's been done for elephants, it's been done for buffaloes, it's been done for kudus, but it hadn't been done for everything. There's another way we can do this, okay? And I'm going to make the proposal right now. Here's another way to do it. It is quick, it is not expensive, it is powerful, and it is doable. So stay with me just for a minute and let me explain what this technology is and what I'm proposing we leverage. This more affordable way to develop these technologies for exotic species could involve the use of a resource that was paid for by the USDA now 15 or so years ago. Some of you that raise cattle know about this tool. It's called the Bove SNP 50 version 3 chip. This is a genome-specific chip that was made for cattle. It very nicely identifies mutations in cattle. 50,000 of them are on this chip. It is used for all kinds of purposes now: breed identification, muscle tenderness, lipid profiles, fertility, all kinds of reasons that this chip is used in cattle. It's very, very routine. Tens of thousands of cattle a year are genotyped with this chip, okay? This is a very common chip. It has been run on some wildlife species. There are some pretty good examples. I'm going to give you one here in a minute. It's cheap, it's cost-effective, and it's powerful. Now, we're not going to get 50,000 markers from an impala. This is a cattle-specific chip. But we will get enough markers to be able to make inferences about what's going on, particularly in things like parentage, particularly in things like breed identification, and very importantly, for individual identification. We can use this chip to develop a DNA fingerprint for your animal. If you have a kudu, like that guy right there, it's a valuable animal. If something happens to that animal and he disappears, you have a microchip in him, you have an ear tag in him, you have a tattoo on him. All those things can be changed, altered, but his genetics cannot. If you had a DNA fingerprint of that animal and something happened to him, he disappeared, and you found him or you found parts of him, you could identify using DNA that it came from that animal. It's my view, if you own animals, you ought to have the opportunity to have them DNA fingerprinted. It's cheap, it's permanent, it doesn't change, and if I was underwriting your insurance, I think I would insist on it. Here's a study that was fairly recently done using this bovine SNP 50 bead chip on white-tailed deer. We have a lot of other resources for white-tailed deer, but most of them started with this bovine chip. So 50,000 markers in cattle. In bovids, they jumped over to cervids. So we're making a huge taxonomic jump here from cattle to cervids. But still, more than 1,000 of those nuclear markers worked in whitetails. And it is a beautiful way to do parentage in whitetails, breed identification in whitetails, subspecies identification in whitetails in some cases, and individual identification, or looking for inbreeding. If you're interested in, if you think you have an inbreeding problem in your herd, you could use this chip with whitetails and it will tell you, it will estimate for you the amount of inbreeding in that animal. So it's been done, and it's been done very successfully. I propose that we need to start a conversation, and we need to start a conversation where we decide if we want to do this or not. I'm interested in it. I've got the wherewithal, I've got the laboratory, I've got the interest. I just don't have the money. Now, I'm not asking you guys for the money, but I'm asking you guys to help me find a way to do this. We can't go to the USDA. The USDA is not going to give us the money to develop this, even though we're not talking about a huge amount here. We can't go to National Science Foundation. We can't go to National Institute of Health. We probably can't even go to Parks and Wildlife. I don't know about that, but I think it would be limited. Where would we go to get the funds to do this? There are foundations, there are places to get help to do it. And two, we're not talking about hundreds of thousands of dollars to develop this resource. We could do it from as few as 10 unrelated animals in the US and 10, as far as we can tell, unrelated individuals at their parent native population source. We can't just do it from the animals in Texas or in the US. We need to do it worldwide for these species. Genotype these animals on this Bove SNP 50 platform, characterize and develop markers that are unique to each species, using the same tool for all of these species, same chip for all these species. The developmental cost for this, is really not a lot of money. Getting the DNA samples, isolating the DNA, characterizing the DNA, running it on the chip. For each of these individual species, we're talking about probably $5,000 in developmental money. Subsequently, to run these animals on the chip, it's going to cost between $25 and $50 to run this chip for these animals, to get this information and to get it analyzed, because it does take someone, a bioinformatics person that knows what they're doing, to be able to analyze this and tell you if there's inbreeding or tell you what parentage information you need. I think the benefits to the exotic wildlife industry are permanent species identification. I think that's important. Documented parentage determinations, I think that's important too. And genetic diversity and inbreeding values can be calculated from this kind of data. So what I've tried to lay out here for you is a plan that is not terribly expensive, that would be powerful enough to get you down the road, and would provide an opportunity for there to be a place where you could send your samples to have this done. Right now, there are companies around, DNA Solutions in Oklahoma, University of California at Davis for some species, other veterinary schools around the US, or you could send your stuff, I guess, to South Africa, because there's some great labs there that are doing some of this kind of work. But the thing is, there's no easy place, at least in Texas, to get this done. And I think Texas A&M is the perfect place to do it. And my lab's got about 30 years of experience with genotyping livestock, bison, and other species. So, am I here to try to get a lot of work? Am I here to try to do a lot of parentage testing, individual identification for you guys or any group of people that has exotic wildlife? And to tell you the truth, no. That's not my major objective for coming here. Remember, I'm a geneticist. I do genetics research. This right here is really not research. This is a service component of what I would do. This would be like you taking your dog to the College of Veterinary Medicine to the small animal clinic because it's got something wrong with it. Okay, that's a service. That's not research. That's a service that we do. Parentage testing, breed identification, inbreeding, that's a service. It's not research. I'm really interested in the development of new knowledge. That's what gets me up in the morning. Being able to learn things that we don't know before. And I can't do that as a geneticist without DNA samples. So remember that WinCabe project where I had 10 years worth of calves, and I made an argument to the Department of Interior, give me the money, let me do this project, I already have the samples, and I have 10 years worth of samples. That's the only reason I got the money to do that research. I am very interested and serious about the DNA samples, because if I have DNA samples, and I have your permission to use them, And you're not identified anyway, so we wouldn't do anything that would impact your operations. But if we have DNA samples from bongos or from other things that are really hard to get, then we can propose to various places to do research. The research I'm really interested in right now, genetic research, involves genes that are involved in the immune response. We have some tremendous opportunities with mRNA vaccines that were developed in the last few years. I'm not interested in developing the vaccines, but I am interested in using some of the technology that came out of COVID-19 vaccine research, looking at genes and how those genes allow those species to respond to infectious agents, and why wildlife don't respond to vaccines made from cattle the way cattle do. We don't have any on-label vaccines for most wildlife species. It's all off-label use. And trust me, brucellosis vaccines don't work in bison like they do in cattle. And the other vaccines don't work in bison like they do in cattle. And right now I have a project looking at why is that the case. And I have to have the DNA samples to be able to do that research. So that's my motivation. So I laid out kind of a vision as what I see we could do. I think it would benefit this industry quite a bit. And I know it would benefit me and my research and other researchers at Texas A&M and probably around the world if we had access to your DNA samples from the animals that you have, because you guys own some incredible animals. And having not just the DNA sample, but having the metadata that goes with it, parentage of that, what the offspring were like, what the animal's health history was, what its body characteristics were, and so forth. So thank you guys. Thank you very much for letting me talk about genetics and not having to think about giving that final on Tuesday to those students. Thank you.
Thank you, Dr. Derr, for, again, a remarkable presentation that hopefully is inspiring some thought. And I think that, going back to what our motive is here in doing all this is that we're really hoping to trigger your mind and encourage you to start thinking about the opportunities to do things better, to improve, to impact our industry in a positive way. And so, one of the things I wanted to touch on just about what he brings to the table and how it directly affects us. If you look at where we are as an industry right now, and I'll talk about this a little later in my presentation, and I equate this to the gold rush of 1849. The industry's been around a long time, but as the result of a lot of changes that have happened in the last 6 or 8 years, there's this massive influx of capital coming into our industry. And it's not coming from people that don't have financial resources. It's coming from people that have really deep pockets. And when you look at kind of what goes on that affects the market, I'll use a few examples. If you look at Nubian ibex, Trans-Caspian urials, mainly sheep and goat species, where you have lots of hybridization, and you look at the economic performance of these species, they've not only become stagnant, but they've gone backwards. Hybridization has affected the financial performance of these particular species, and as the result of that, they've become less desirable to many people. And so, why is that relevant? Well, it's relevant when you think you're buying something pure. You see a Trans-Caspian urial, it's got all the look of what you're after. It's got all the characteristics that you want. It's got this great quality bib that's on its front. It's got the horn structure. But are you actually buying a pure animal? Are you actually buying an animal that is in fact what it's represented to be? And with samples that we provide to Dr. Derr, over a period of time, he has the ability to answer these questions for us. We're starting to see species like Simbach, evolve, and are these animals actually capable of breeding on a consistent basis? I've seen at auction where an animal is being sold as a Gemsbach, and it is in fact a Simbach. It's just happened to be bred back and bred back and bred back over time to where it's almost indistinguishable but it is absolutely indistinguishable to someone that's just getting involved in the industry. So if you're just getting started and you're in an auction and you see a animal that runs through the ring and everybody that's been doing this for a while knows it's a Simbach, and you don't, and you pay $10,000 for it thinking it's a Gemsbach, and you walk out high and mighty thinking you just got a great deal, well, you just bought a hybrid. And these are the kinds of pieces of information that we're trying to share with the industry because they directly affect the stability and the transparency and the trustworthiness of our industry. And so there's nothing I like more than data that proves beyond any doubt whatsoever the facts around what it is that we're doing. And Dr. Derr brings a tremendous opportunity and a tremendous resource for us. But the last thing I want to touch on in that is that he mentioned what's coming and what is in fact going to happen. You know, we're seeing embryo transfer work being done in species. We're seeing very, very deliberate and intentional breeding going on within various species. And when you look at the people that are involved in this and you consider their financial resources and what they have the capability of funding, there's some really, really incredible possibilities that are in front of us that can be great or they can be not so great. And I think that when you take into consideration that there is this underlying financial motive that comes with what all of us are doing, if you spend a $500,000 to develop something that ultimately is worth $5 million, well, it's kind of a no-brainer. And there are going to be people over the next few years, I think it's a shorter horizon than a longer, but there are going to be people that are spending the money to do really important and really significant genetic research, as well as various types of breeding practices. And so if you want to be a part of that, the opportunity is to do it, or if you want to be a bystander, you can be a bystander. But I think you're going to see here over the next few years, as you're evaluating what you're doing from an investment standpoint, take into consideration that these really, really, really technical, scientific approaches are coming. They are going to happen. Kevin and I, you know, we look at Cyclone up here. Well, Cyclone is not young. He's elderly, and we have to give him a rest because we've got him on 20 or 25 cows, and he's getting a little bit weak in the backside. And so we don't know how long he's going to live. So we've talked about drawing semen just simply to be able to give us a guarantee that we believe this might be the largest kudu in the world today. And so I mentioned to him, well, let's look at cloning him. You know, and when you start talking about, well, he says I didn't say that, but I did say it. Okay, well, he says it won't work. I'm going to get Dr. Derr to say it will. So anyways, the point is that today we think things may not work, but tomorrow we may find out that they do. And so the point is people are going to try. They're going to try. The money is here to be able to do this. And so I encourage you to get with Dr. Derr. Let's get him hair samples. If there's funding that needs to happen, let's get the funding. There's no reason that as an industry we can't take the lead on helping these universities to do the research, to advance what it is that we're doing, and to create a pathway that is really based on things that are factual, not gossip, not let's just take a guess and hope for the best, but let's spend the money to do the research because I think it'll benefit our industry tremendously. So thanks again, Dr. Derr. Really appreciate you being here.